Technology. তোমাদের কি শুরু করেছিলাম এখান থেকে কিছু? নো, আই থিংক
না, আমি শুরু করা হয়নি।
আমাদের টাইম খুব কম। এখন তো দেখি নেক্সট কোয়েশ্চেন ফাইনালের আন্ডারগ্রেডের জমা দেওয়ার টাইম চলে আসলো। So anyway, from now on in the final syllabus we will be studying mostly about the application part, like with recombinant DNA technology, PCR technology, and sequencing. We learned more about the techniques that are used, but we haven't gone into large scale production of something. And biotechnology, obviously, it involves large scale production in industry. So that thing will start from tomorrow, and we refer it with fermentation technology. Now, fermentation word, if you think of fermentation, simply you have studied in school what it means. It's some kind of anaerobic process where the end product would be further oxidized. Like something is formed, it's not the completely oxidized product. You can still oxidize it. Like, say, for example, in a ethanol fermentation, when ethanol is produced, ethanol is the product that would be, this is alcohol, this would be further oxidized into aldehyde, into acid, and final oxidation that leads to formation of carbon dioxide. So classical definition of fermentation is without air. So anaerobe redox reactions in organisms, that was termed as fermentation. In school we read in this way. But in case of fermentation technology, as far biotechnology is concerned, this simply involves the development of new fermentation processes. Now, what fermentation processes? The process could be aerobic, it could be anaerobic. It simply means large scale production, large scale culturing. So here you see, for all practical purposes, fermentation in case of biotechnology, this is generally defined as the process of growing a culture of microbes in a nutrient media, converting the substrate or feed into your product. So the definition of fermentation changes with reference to the context. So in industrial biotechnology, you will always refer it. It's not that process should be anaerobic. It could be aerobic as well as anaerobic. But what we are doing here, large quantities of the cells are grown or product is formed through conversion of a substrate into a product. Now, throughout the fermentation technology, we understand that fermentation now it means you will culture organism, that organism will convert the substrate into the product during the growth process. Now, the problem is, all of you have seen in laboratory when you culture microbes in test tubes or shake flasks, conditions how we manage. Simply say, for example, it is aerobic growth in E. coli. Say in lab when we grow E. coli, what we do in test tube or conical flask, we don't fill the whole vessel. Like if it is 10 ml tube, we don't fill it with 6 ml, 7 ml, 8 ml of culture media and then we put E. coli. No. Why not? Can anyone answer? क्यों बोलते? Why don't fill up the tube? Because if we fill up the tube, there the oxygen is coming from air. And if you fill it up, the head space, the space above the tube or the flask, that will be occupied, so oxygen demand will not be met. So usual idea is for, the good assumption is 1:10. So if you have 10 ml test tube, you will put 1 ml culture media into that. If you have 250 ml flask, you will go 25 ml culture media into that. So why this is done? Because we provide air. But when you go for industrial production, you can't say 40,000, 50,000 liter of culture will be produced. You can't make a vessel like 10 times of that. That will be quite expensive. So what you need to do, you need to provide the controlled conditions with some mechanical intervention. Like for mixing, you have to there in laboratory what we do for mixing, we simply put the test tube and flask into a shaker. শেকারে দিয়ে দিয়ে মিক্স হবে মিক্স কাকে করতে হবে there are three phases in the media যেখানে গ্রোথ হচ্ছে there are three phases solid liquid and gas solid কোন পার্টটা বলো তো যখনই কোনো অর্গানিজম গ্রো করছে which one is the solid phase কি কেউ শুনছো
জি মিস
তো আনসার তো দাও সলিড ফেজ কোনটা হবে লিকুইড কোনটা হবে লার্জ স্কেল প্রোডাকশন যখন হবে লিকুইড পার্ট কোনটা হচ্ছে the media itself culture media liquid and the solid phase is the cell itself cell biomass microbe je grow korche that is the solid phase and gaseous phase is the air air okhane jeta ache that is providing the oxygen so these three phases should be mixed jate nutrient transfer mean mass transfer as well as heat transfer proceed adequately. So to provide those controlled conditions, we need specialized vessels. Those are for large scale production. Those are called fermenters or bioreactors. And we also need to optimize those conditions. We have different ways. You may choose different type of mixing devices. You may choose different type of mode of operation of fermenters, how they will operate. So those things you will see that what type of fermenters are available. So, so far, although classical definition of fermentation is anaerobic, but most of the biotech drugs, if you see, they are grown with aerobic fermentation. So what is the difference between aerobic and anaerobic fermentation? As the name suggests, aerobic fermentation that requires aeration. Aeration, why? Because it will fulfill the demand of oxygen. So the bioreactors or fermenters that you have, they must supply adequate amount of sterile air. Here, the air that you will provide, this should be sterile. আমরা যখন ল্যাবে করছি in test tube and flask what we do আমরা ওই test tube এর মধ্যে ওই either ওটাই sterilized থাকে or ওই test tube এর মধ্যে or ওই flask এর মধ্যে media নিয়ে sterilize করছি assuming the air inside is also sterilized but here when we provide the air from outside that air should be sterilized otherwise it will contaminate your product and sterile air how air could be sterilized কে বলবে এয়ার কিভাবে স্টেরিলাইজ করব এনিওয়ান
HVAC
air could be sterilized using HEPA filter usually HEPA filter is used mean filter sterilization is done and together with this you must have a mechanism for stirring and mixing so example of the products that you have using aerobic fermentation this includes antibiotics enzymes vitamins hormones and lot of biotech drugs they are mostly made with aerobic fermentation so where anaerobic fermentation is used anaerobic fermentation is mainly used in chemical industry where bulk of chemicals are produced like lactic acid ethanol wine in those cases anaerobic fermentation is required so idea for fermentation is why you are growing those cells to convert a substrate into a product now if you grow the cells any bacteria or any other organism say yeast or any other organism you will provide it with media so that it can get the nutrition but together with nutrition It must have that substrate that will give you the product. For example, if you want to make curd, hmm, we know which bacteria say lactobacillus. You can't provide it in water and expect bacteria will grow. Say nutrient broth you are using. Nutrient broth is culturally rich media. There also those organisms will grow or other organisms will grow. But this is not going to give you curd. So that means your media should be such that it contains the substrate that will give you the product that you need. So media should be designed carefully. That includes the substrates. And when you inoculate that media with the cells, the organism that is going to convert it, you inoculate that, and that will lead to growth of those cells. So growth is called biomass. Cell will multiply and increase in number, and during the process of growth they will form the product. And the product could be your desired product that you need, but apart from that other products will also be formed. Those will be metabolic waste of the bacterial system or any other system. So waste products will also be formed together with this. As this is metabolic process, with metabolism, heat will also be generated. Large amount of heat will be generated. Hmm. So different type of cells could be used. So during the recombinant DNA technology also we discussed that as host we mostly choose bacteria. Why? Because it's easy to grow and cheap. Hmm. You don't need to have very expensive facilities to grow bacteria. It's easy to extract the product and purify the product from bacterial culture media. So our first choice is usually bacteria. So in case of bacteria also, it's a E. coli is very well known. It's genomics studied. E. coli, bacillus, pseudomonas, streptomyces. Like pseudomonas is used for vitamin B12 production, streptomyces for antibiotic production. So those are there. Then yeast. Yeast can also be used for insulin production as well. Together with this, like Saccharomyces species, you can use that's baker yeast, etc. Mold, penicillium, and নাম তো সবাই জানো for making penicillin. Aspergillus, like Aspergillus niger, is used for making citric acid in chemical industry. আর যখন এইগুলো দিয়ে হবে না, then we go for higher cells. We have studied in gene expression analysis that some cells require these type of modifications, some products require. So those type of phenomena, they don't happen in bacterial cells or yeast cells. Then you go for other type of cells. So animal cells, if you choose mean you have to choose, say, for any reason, the product will become expensive. Why? Because animal cells growing is not an easy thing. Easy thing mean it's not cheap also. Culture conditions also thing. You need to have acidity control should be there. So everything requires lot of expenditure. So in case of animal cells, here some examples are given, like Chinese hamster ovary cell line, CHO. Simply NIH cells, NIH3T3, that is mouse cell line, that is also used. Hybridoma cells, you are aware that this is used for monoclonal antibody production. And then insect cells, they are also used for certain type of protein. Together with this cell, এখানে আমরা isolated cell এর কথা বলছি, but together with this, as we know in bee farm, we also use for biotech example animal farming যে animal suppose milk production হচ্ছে, ওখানে কোনো একটা জিন আরো ইনক্লুড করে দেওয়া হচ্ছে, so that product is also produced along with the milk. Similarly, in case of plant cells, plant cells also প্ল্যান্টের মধ্যে কোনো একটা প্রোডাক্টের জিন ইনক্লুড করা হয়েছে with the plant growth, and that could also be an option. So these type of things you will be using for fermentation. Now here you see the picture of a small scale fermenter. So fermenter, and another term that is frequently used is bioreactor. So what is the difference actually between fermenter and bioreactor? These are usually nowadays used mostly, but there is a technical difference. Fermenter is usually, we when we grow microbes, we call the vessel as fermenter, but when we grow plant, but nowadays mostly they are used the same term, so any could be used. So if you see the overview of the whole fermentation process and plant, what happens, you need to first procure all the raw materials. Next you need to process those materials. So that processing, before fermentation, the processing that you do, that is called upstream flow chart. And of the mouth, again with the help of a flow chart, and then finally the purified product goes here, that you will supply to pharmaceutical companies who will formulate that into doses form. So now we will go to different stages of fermentation: upstream processing, fermentation, and downstream processing. So just go with this flow chart. It has everything shown here. So upstream processing, first thing that you see whenever you go for any fermentation process, it's like a formulation also. Whenever you make a formulation, say of paracetamol, which one you will mention, which ingredient? Hmm, কি কেউ বলছো না? In pre-formulation,
এপিআই গুলো
right, API ওখানে study করা হয়. So here also, like in case of fermentation, your primary doer is whom? The organism. That organism will convert the substrate into the product. Say for example, if you have to make insulin, of course you need to use that genetically engineered E. coli that is able to produce that insulin. So first thing and the most important thing is you have to choose the right microbe that will make the product for you. So first thing you see here, it's given microbe selection. You have to isolate the microbe. Isolation কখন করতে হবে? For example, if you are developing something new, you need to isolate, or maybe if it's already ongoing type of thing, then you may already have that isolated strain. In that cases, you only need to maintain the strain purity. But even if you have that isolated strain, always remember in this type of industry, you continuously go through research process. The strain that you have, it should be continuously improved. কেন আজকে যেটা আছে 10 বছর পরে টেকনোলজি একই রকম চলবে না. The organism, better organisms will be coming with better yield value. So you need to improve the strength. For strain improvement, again, there are many different type of ways, like further genetic engineering, different type of mutations, change in media, change in growth conditions, change even pH, etc., whatever is required, or substrate also. So strain improvement. And strain improvement, when you get the desired strain, it will go into production. So your production strain is ready, and here you see the another arrow is shown. The production strain also undergoes through research for further improvement. After few years or after some time, you may have a better variety. Now, when you have your microbe ready, the strain is ready, you have to study it carefully for its requirements. Like in pre-formulation study, you study all the characteristics of the drug of which you are making that formulation, because based on that only you will choose other excipients. For excipients, you have other alternatives. You have many alternatives. So what you will do? First, you need to study the API. So here what you need to study, the microbe itself. So first you have to grow it at large scale. So you need to understand what it needs, what are its nutritional requirements, what are its metabolic controls, what is the optimum pH temperature that it requires for growth, oxygen, carbon dioxide কত লাগবে, morphology কিরকম. Some organisms have this filamentous growth. So if that type of thing is happening, what will happen? It will interfere with mixing of the ingredients. So you need to think whether foam is generated or not during the growth. Genetic stability, especially if the organism is genetically modified organism, it's important that it doesn't lose that plasmid or doesn't lose that gene. Otherwise, your product will be not coming. If insulin gene is lost from E. coli, obviously you will not get insulin. Apart from this, what kind of metabolic byproducts are formed? Why byproducts? Because some of the product that may be formed, it will be very difficult to eliminate them. They could be toxic as well. So what type of other products apart from your desired product is getting formed, that also you need to know. Viscosity, flow properties of the culture media that will be required in the later stages of purification. So all those things you need to study first. What is the requirement of that organism? So clear so far? So next is what? Once you know what it requires, it will require carbon, nitrogen, oxygen, sulfur, jaja lage, trace element, growth factors, water, etc. Now, if you see, carbon sources are many, nitrogen sources are many, phosphorus sources are many, so there are many options for you. Now, media, when you design, there are again different types of media, like complex media, defined media. So what type of media you will choose here? Complex media is what? It contains the complex source of nutrition. Complex media doesn't mean that making is complex. Defined media mean defined media mean all the components of that media is known. Like this much amino acid is there, this much vitamin is there, this vitamin B12 is this much, vitamin C is this much, alamine, arginine is this much. So all the components will be defined. Calcium chloride, this is this much. But in case of complex media, like you prepare in your lab nutrient broth, those type of media, they are complex media. Why? Because there we use beef extract or yeast extract, tryptone, peptone, thinking that this will fulfill all the nutritional requirements of the organism. So that means it is a complex source of nutrition. For example, when you are taking your food, you have rice and dal and those things. So they are complex source of nutrition. They contain all the things, those amino acids, those vitamins, etc. But we don't know the exactly এত amino acid থাকবে, এই particular amino acid এত amount থাকবে, এভাবে আমরা করি না. So Here, what type of media you will prefer to grow that organism? Defined media or complex media? Can anyone answer? What would be better? Defined media? Defined media. Okay, not defined media. Why? Because defined media, তুমি ধরো তুমি নিজে খাচ্ছ, you have isolated vitamin D এতটা, vitamin B এতটা, vitamin C এতটা, this amino acid this much and this is this much, and you are taking complete food আরেক জায়গায়, দুটোতেই তোমার গ্রোথ হবে, বেটার হবে, or maybe ওটাতে আরো বেটার হবে when you are taking complex source of nutrition. So idea is, and of course that will be cheap. The complex source of nutrition, complex media will be much cheaper. You don't, if you get purified amino acid, purified chemicals, trace elements, everything, the cost will be very, very high. Individual product you need to have in purified form. So that will be difficult. Here your objective is what? To grow the organism at large scale. If the organism grows properly in that media and convert that substrate into product, why you will go for defined media? Because this will simply increase the cost. বুঝে গেছে? Yes, ma'am. So together with the availability, যে তুমি এমন মিডিয়া চুজ করেছো, like sugarcane industry jein product molasses, that is also used in fermentation industry. Corn steep liquor, that is also used in industry. But the thing is, whatever is available locally, তুমি দেখেছো আমেরিকাতে একটা প্রোডাক্ট এইভাবে করছে, but that raw material is not easily available at your place. If you import that raw material simply to grow the organism, your cost will be much higher. So what you should see for that, here what is easily available to you, that the microbe can utilize to give you that product. So availability, cost, stability, and pre-treatment and sterilization. All the media, whatever you use, that should be sterilized. And pre-treatment mean if you are taking some crude form, whether it could be used in that form by microbe or not, that you need to see. If it is not usable, you need to pre-treat, say for example, কিছু enzymatic hydrolysis to break down some of the products so that microbe can utilize it easily. So whether there is any requirement for that or not. So based on this, you will choose which raw material for media preparation you'll be using. Now, once you have chosen the composition of media, next is, here you see propagation media, maintenance media, and then starter culture propagation এর জন্য একটা media, that is propagation medium, and this is the production medium. তো এই তিনটা মিডিয়ার মধ্যে কি ডিফারেন্স আছে? So once you develop the media, production media is easy, যেটা তোমার ফার্মেন্টারে যাবে, that is the production media. Their large scale production will be happening. Maintenance media is what? You will save it for future use. আমি এই মিডিয়াতে অর্গানিজমকে মেনটেন করব for next batch or maybe for years, whatever you want. So in case of maintenance media, the difference will be simply what? You will choose the type of substrates that will be slowly metabolized. Or maybe যেমন খুব common example in case of lab work, if you see, when you have to preserve or maintain any cell, we choose solid media. But if you want to grow at large scale, we choose liquid media. So why? Because in liquid media, growth will be faster. In solid media, for 15 to 20 days, you may preserve, because colony will also be single there without any contamination. Together with growth will be very slow. So maintenance media may have slowly metabolizable product so that organism remains viable. In case of liquid media, the growth will be very rapid and the death will also be rapid. So the organism will die within few days. So we choose a special maintenance media. And the propagation media, here you see, this is actually more or less similar to production media. Propagation media আমরা কি জন্য ইউজ করছি? All of you have given me an introduced organism into culture media. That process is called inoculation. When you inoculate bacteria into a culture media, what we usually do, we sterilize the loop, we pick the colony, and we stir this into the media. That is called single colony দিয়ে আমরা bacteria নিয়ে inoculate করছি. But here your culture is say 50,000 liters and like that, large scale culture. Here if you give that single colony inoculation, the lag phase, the bacteria will take long time to multiply, and the lag phase will be increased, and that will lead to unnecessary cost burden. So what we need to do, we need to first prepare a starter culture. হেলদি লগ ফেজ সেল আমরা আগেই গ্রো করে রাখবো, দ্যাট মিডিয়া ইজ কল্ড প্রোপাগেশন মিডিয়া, যে মিডিয়াতে আমরা গ্রো করাবো। নাও ইউ মে আস্ক হোয়াট ইজ দ্য ডিফারেন্স বিটুইন প্রোপাগেশন এন্ড প্রোডাকশন মিডিয়া। অ্যাকচুয়ালি প্রোপাগেশন মিডিয়াতে আমরা শুধু গ্রোথ করাচ্ছি, এখানে ওই সেলকে আমরা একটু হেলদি লগ ফেজ সেলকে নাম্বারে বাড়াচ্ছি, বাট হিয়ার উই আর নট প্রোডিউসিং দি প্রোডাক্ট। In production media, you may have some type of substrate that will actually increase the production rate. Maybe jodi gene expression hoy, say for example insulin, you may like to add say inducer IPTG or XGAL like that. So that will be required for production media, not for propagation media. So these are some minute differences between different types of media. So starter culture you have produced, and the amount is usually fixed, that how much starter culture you need to give in how much production media. So once you have inoculated that into production media, now remember when you put the media into here, in case of fermenter, the media will be sterilized within the fermenter itself, because in lab what we do, we sterilize the media in the flask or test tube, etc. And whenever you require, you may transfer into some other sterilized vessel. But of course, here you can't sterilize 50,000 liter in some autoclave and again you put here, and your fermenter should also be sterilized. So in these cases, provision is there for sterilization of the media within the fermenter itself. So fermenter will also be sterilized together with the media. So for that, boiler steam এখানে provision থাকবে, আমরা fermenter design যখন দেখবো, you have the provision to supply steam যেটা দিয়ে autoclaving হবে। তো together with this you need to take care of oxygen, এখানে দেখতে হবে অক্সিজেন ডিমান্ড কি রকম আছে, সবই কিন্তু ডিসপ্লেতে আসবে। এর আগে একটা এখানে যে পিকচার দেখছো, এখানে ডিসপ্লেতে কিন্তু সবই থাকবে, এখানে দেখিয়ে দেবে অক্সিজেন লেভেল কত আসছে, পিএইচ কত আসছে, because when the growth is happening at large scale, it will change very rapidly. It won't be like it happens in lab যে আজকে দিলাম কালকে growth দেখছি। In large scale growth, you need to monitor it very closely. So for that you have those display, যেখানে তুমি দেখতে পাবে পিএইচ যখনই বেশি গ্রোথ হবে, acid will be produced, pH will be down. When you see the pH is getting low, you need to provide with say sodium hydroxide or something to maintain that pH. So everything will be there, oxygen control, pH control, anti-foaming agent. Large scale production যখন mechanical mixing হবে, stirring হবে, ঘুরবে, it will cause formation of foam. ফোম যদি খুব বেশি প্রডিউস হয় তাহলে কি হবে, মাস ট্রান্সফার, এই যে তিনটা ফেজ বললাম, সলিড, লিকুইড, গ্যাস, এই তিনটা ফেজের মিক্সিং কিন্তু প্রপার হবে না, ইনহিবিট হবে, বিকজ মাঝখানে এয়ার বাবলস গুলো চলে আসবে। সো ইউ নিড টু প্রোভাইড, যদি বেশি ফোম প্রডিউস হয়, ইউ নিড টু প্রোভাইড অ্যান্টি ফোমিং এজেন্টস দেয়ার। এই বাদে কুলিং, হিটিং এর প্রভিশন থাকে। ইউজুয়ালি ফার্মেন্টার গুলো জ্যাকেটেড থাকে, যেটা দিয়ে তুমি হট ওয়াটার আর কোল্ড ওয়াটার ফ্লো করাতে পারো, সো দ্য টেম্পারেচার কুড বি ইজিলি কন্ট্রোল। যত তোমার বেশি অ্যামাউন্টে মেটাবলিজম হচ্ছে, লার্জ স্কেল কালচারিং হচ্ছে, হিট উইল বি প্রডিউসড, এন্ড ইট উইল বি সো মাচ দ্যাট তুমি যদি টেস্ট টিউবে করো, নেক্সট ডে তুমি যখন টেস্ট টিউব টাচ করে দেখবে, ইট উইল ফিল মাচ ওয়ার্মার। কিন্তু এটাই যদি আরো লার্জ স্কেলে হয়, তাহলে অবভিয়াসলি temperature increase will be faster. Now, if say E. coli grows at 37 degrees centigrade and your temperature has reached to 45 degrees centigrade, obviously the growth will be hampered, product formation will also be hampered. So there is a provision for cooling as well as heating. So this was about upstream processing. The upstream processing, microbe selection, media preparation, and then you will move towards fermentation. So after fermentation, how long you will proceed with that fermentation? That means you will give 24 hours to fermentation to finish, or 48 hours, 72 hours, how much? So that depends on when your product will be at the peak level. So how will you determine that? We will study with growth phases. Now, once if it is finished, fermentation, say 24 hour, 48, joto time lagbe, you're done with fermentation. That means product is formed there. You stop the fermentation process, and then proceed for purification and isolation and purification of the product from the fermenter. Kono question ache karor?
ম্যাম, ম্যাম প্রোপাগেশন মিডিয়া আর হচ্ছে প্রোডাকশন মিডিয়ার ডিফারেন্সটা আরেকটু একটু বলবেন?
Propagation and production. Propagation media is simply for growing the cells, healthy log phase cells. So growth যেখানে লাগছে, only nutrients are essential. But when you go for production means that specified compound that you are producing. The example দিচ্ছিলাম, like say you want to produce insulin. তো ইনসুলিন জিন আমি ওখানে ধরো ক্লোন করেছি, আমি কি চাইছি, ইনসুলিন জিনের এক্সপ্রেশন বাড়ুক, যত বেশি বাড়বে তত yield বেশি হবে. So in that case what you need, maybe you will also add an inducer like xgal IPT যে আমরা পড়েছি, যে ল্যাক্সি জিনের এক্সপ্রেশন বাড়াবে। তো ল্যাক্সির সাথে আমাদের ক্লোন কি আছে, ইনসুলিন জিন। তো ওই মিডিয়া গুলোতে আমরা এমন জিনিসও অ্যাড করতে পারি যেখানে এক্সপ্রেশন বাড়বে, কিন্তু যখন আমি প্রপাগেট করাচ্ছি শুধু আমার সেল দরকার, I don't need the production of that particular insulin. So in propagation media, I don't need to add inducer and those substrates. Is that clear?
Yes, ma'am.
So next thing is how to isolate and purify the product. So isolation jodi boli, how you will do? Fermenter যেটা ছিল ওখানে প্রোডাক্ট ফর্ম হয়ে গেছে, ওখানে মিডিয়াও কিছু থাকবে, তার সাথে প্রোডাক্ট থাকবে, and your product could be intracellular, that means the product is accumulated within the cell, or এমন হতে পারে your product is extracellular, that is secreted in the media. দুটো কেসই হতে পারে. So first thing that you need to separate the cell from the media. Cell আলাদা করব, biomass and media আলাদা করব. How you will do? একটা তো ওয়ে হচ্ছে filtration করা through bacteria proof filter. তো আমরা যেমন ফিল্টার স্টেরিলাইজেশনে, মেমব্রেন ফিল্ট্রেশন এটসেট্রা করেছি, কিন্তু যখন আমরা ইন্ডাস্ট্রিতে অ্যারাউন্ড এই 50,000 লিটার এইরকম কালচার নিয়ে কাজ করব, we need large scale devices, equipment যেগুলো ইউজ করব Maybe you have studied, maybe now you have studied in pharmaceutical engineering as well, like plate and frame filter press, rotary vacuum drum filter. তো এই টাইপের ফিল্টার্স গুলো এখানে ইউজ হবে, যেগুলো আমরা পরে একটু ইনস্ট্রুমেন্টেশনে দেখবো। তো either you go for filtration or you go for centrifugation. So centrifugation, simply the heavier objects will settle down during centrifugation process. The cell কিন্তু easily separate হয়ে যাবে even if you rotate it at 5000 rpm for a few minutes, 5 minutes, 10 minutes, like that. So centrifugation is one of the big. So for centrifugation also, you won't be using here the lab scale centrifuges. Lab scale centrifuge সবাই দেখেছো, ওখানে 10 ml 50 ml এইভাবে আমরা সেপারেট করি। তো এখানে আমরা যে সেন্ট্রিফিউজ ইউজ করব, দ্যাট ইজ ইন্ডাস্ট্রিয়াল ইকুইপমেন্ট। একসাথেই একটা বোল টাইপের থাকে, ওর ভিতরে ওটাই ঘুরবে, ওখানেই সেন্ট্রিফিউজ হবে। তো ওগুলো আমরা দেখবো কোন টাইপের ইনস্ট্রুমেন্ট ইউজ হয়, বাট দ্য প্রসেস ইজ either centrifugation or filtration. তো এটা দিয়ে আমরা কি করব, সেল আলাদা, মিডিয়া আলাদা। Now which one you will take? If your product is intracellular, you will take cell, you will discard media. Discard mean এমনি ফেলে দেবে না, প্রপার ওয়েতে ডিসপোজ করতে হবে। And if your product is extracellular, you will discard cell, that also you will discard in proper way. Now think if your product is intracellular, like in case of insulin, insulin is within the E. coli, it's accumulated within the E. coli. So if the product is intracellular, you take the cell, এখানে দেখছো লেখা আছে intracellular or periplasmic product. Periplasm কোন পার্টটাকে বলা হয় জানো? Anyone? পেরিপ্লাজমিক হচ্ছে, আমরা মাইক্রোবায়োলজি মনে ভুলে গেছো, গ্রাম পজিটিভ ব্যাকটেরিয়া এন্ড গ্রাম নেগেটিভ ব্যাকটেরিয়া দুটো পড়েছিল না? গ্রাম পজিটিভ এর মধ্যে কোন লেয়ার বেশি থাকে? বলবে পেপটাইডোগ্লাইকান। আর গ্রাম নেগেটিভ এর মধ্যে ইউজুয়ালি লিপোপলিস্যাকারাইড বেশি থাকে। তো পেরিপ্লাজমিক স্পেস হচ্ছে বেসিক্যালি, রাইট, গ্রাম নেগেটিভ ব্যাকটেরিয়ার মধ্যে যে স্পেস থাকে বিটুইন ইনার এন্ড আউটার মেমব্রেন, এটাকে আমরা বলি পেরিপ্লাজম। তো এখানে হচ্ছে অনেক সময় অনেক প্রোডাক্ট কিন্তু জমা হয়। তো এই পেরিপ্লাজমিক স্পেস এটা কিন্তু ইম্পর্টেন্ট, কেন ইনসুলিনের কেসেও এই প্রোডাক্ট যে ইনসুলিন, this also get accumulated in periplasmic space. তো পেরিপ্লাজমিক স্পেসও বেসিক্যালি সেলের ভিতরেই যাচ্ছে। তো এখন প্রোডাক্ট যখন সেলের ভিতরে থাকছে, ইন্ট্রাসেলুলার অর পেরিপ্লাজমিক, how you will isolate the product? You have to break down the cell. সেলকে লাইসিস করতে হবে, সেলকে ভাঙতে হবে, লাইক we did in case of DNA isolation. সেলকে ডিসরাপ্ট করতে হবে। তো there are different ways like chemical lysis, enzymatic lysis, mechanical lysis. So you'll choose your method, cell disruption, and after that you will separate the cell debris. Waste material যা যা থাকে এগুলো চলে যাবে। You will get cell free extract or inclusion bodies. Inclusion bodies আমরা বলি যে প্রোটিন তুমি চাইছো এক্স্যাক্টলি সেম ফর্মে না, কিন্তু এটা প্রোটিন এগ্রিগেটেড ফর্মে থাকছে, ডিফারেন্ট বন্ডস আরো আছে যেটার জন্য ওগুলো কানেক্টেড। So inclusion bodies are mainly aggregated forms of that protein. So your product may be in the form of inclusion bodies as well. তো এই অবধি একটা প্রাইমারি রিকভারি হলো যে তুমি প্রোডাক্ট একটা ক্রুড ফর্মে বের করে ফেলেছো। এটা হচ্ছে যদি ইন্ট্রাসেলুলার হয়। আর যদি এক্সট্রাসেলুলার হয়, then what you need to do? In case of extracellular, you need to concentrate the media. মিডিয়া তুমি নিয়েছো, মিডিয়াতে যদি তোমার প্রোডাক্ট থাকে, obviously it will be very diluted. কেন ধরো 50,000 লিটার যদি এক্সাম্পলি ধরি, তুমি যদি সেপারেট করে ফেলো সেল বায়োমাস কে, অ্যারাউন্ড ধরো কত লিটার যাবে? 2000, 3000, 4000 লিটার, বাকি পুরোটাই মিডিয়া থাকবে। তো এই মিডিয়ার মধ্যে যখন প্রোডাক্ট আসছে, দিস উইল বি ভেরি ডাইলুটেড। কেন, সেল তুমি যখন আলাদা করছো, সেল যখন লাইসেন্স করছো, ইউ আর চুজিং দ্য অ্যামাউন্ট অফ সাসপেন্ডিং এজেন্ট দ্যাট ইউ আর ইউজিং ফর লাইসেন্স। তো প্রোডাক্ট অলরেডি কনসেন্ট্রেটেড ফর্মে আসবে। কিন্তু মিডিয়া যখন ইউজ করছি, the first step that you need to do is concentration. So concentration could be done in different ways. So what you will get here, the concentrated media concentrate. This is primary recovery. Next, what you need to do, এটা crude product হলো, এই ফর্মে তো এটা usable না, so then you need to do purification. Most of the biotech drugs, they will be administered parenterally, like in case of insulin we give injection, and most of them are protein in nature. আর যদি প্রোটিন হয় তাহলে sterilization will be very difficult. আর যদি parenteral product হয়, obviously sterilization is mandatory. You have to sterilize. So, and also this should be highest purity grade. It should be pyrogen free and those things. The purification takes lot of steps, and it also involves lot of money. Why? Because this purification is not done in a single step process. Multiple type of, you will see in case of insulin. Insulin is a small product, but you will see how much purification stages it goes through. So there are different ways to purify a product, like dialysis, precipitation, partitioning, different type of chromatographies, ultrafiltration, distillation. So whatever is required for a particular product, the purification protocol is developed in a particular way. So once your product is purified, এরপরে হচ্ছে আরো স্টোরেজের জন্য, বেটার ফর্মে নিয়ে আসার জন্য, the most purified thing we know is the crystal. That is the identity of the product, the crystal structure. So if you can form crystals, and drying, mostly freeze drying or lyophilization, we do sterile filtration. Finally, you package the product, and this you could deliver to the, after testing obviously, you could deliver to the pharmaceutical industry for further formulation and those things. So here you see fermentation techniques. It could be done as submerged fermentation as well as surface fermentation. Submerged কাকে বলছি, within the culture media, throughout the culture media growth হচ্ছে, that is submerged. আর surface mean like in case of plate, পেট্রি ডিশের মধ্যে যেটা সারফেসের উপর গ্রো করছে। কিন্তু এখানে যখন আমরা লার্জ স্কেলে মেইন করছি, ইটস নট অনলি অন সলিড সারফেস, লিকুইড সারফেসও যদি এমন হয় শুধু উপরেই গ্রোথ হচ্ছে, এরকম কেসগুলো আছে, উই কল ইট সারফেস ফার্মেন্টেশন। আর যদি সলিড হয়, এখানে কিন্তু পেট্রি ডিশ অবভিয়াসলি ইউজ করলে আমাদের লার্জ স্কেলে কাজ হবে না, এই জন্য এখানে বড় বড় ট্রে ইউজ করা হয়। তো দোজ টাইপ অফ ফার্মেন্টার্স আর কল্ড ট্রে ফার্মেন্টার্স। তো এখন আমি একটু ব্রেক দিচ্ছি, অ্যারাউন্ড 10 মিনিটস মতো, তারপরে আমরা ফার্দার ডিসকাস করব what type of fermentation processes are there. Okay?
Okay, ma'am.